Hi all. So hope you are all doing fine. So today we are going to discuss about the much hyped about the antibody cocktail. So my question is it worth the cost and is it worth the hype? And what effect it actually has on the pathogenesis of COVID? So this antibody cocktail, it was developed by the company called Roche, and in India it is supplied by Cipla, and it is a combination of Kesarivimab and the Imdivimab. So the objective of this class is to learn in detail about the mechanism of action of these two antibodies and the pathogenesis of COVID only in respect to these antibodies. I'm not going to go into the pathogenesis of COVID in detail, but only in respect to these antibodies. Okay, so to know the pathogenesis of COVID, you must first know the structure of a coronavirus pretty clearly. Okay, so the coronavirus contains of a nucleocapsid, a spike glycoprotein S, which has two parts, S1 and S2, which we will see in detail later. And there is a membrane protein M, and there is an envelope E, okay, and there is an RNA, and there is heme agglutinin esterase D dimer. Okay, do you get it? These are the basic parts of a COVID virus. Okay, so once you know and you have an idea of the COVID virus, we must see the pathogenesis of the COVID virus. So mind you, we are going to see about the pathogenesis of COVID virus only in respect to how this antibody acts. Okay, so for this I'll make use of a whiteboard for this. So yeah, so let's just draw a picture of this COVID virus, which has just entered the host cell, and it sees a host cell, okay, alveolar epithelial cell. Okay, this is inside the respiratory tract and sees the host cell. What is the first thing it does is it needs to get attached. Okay, it needs to get attached. Okay, for that it has the spike proteins. Okay, this spike protein is S1 and S2. Usually they are bound to each other by a bond. Okay, we have to remember clearly that this S1 and S2 have separate functions. Okay, this S2, it is for binding to receptor. Okay, this S1 is for fusion of cell membrane plus viral membrane. Right? So the next step, what happens is, the first step in a virus getting attached to a host cell is S1 S2 cleavage. Okay, so how does it get cleaved? Okay, how does S1 and S2 get cleaved? There are some membrane proteins on the host cell, like this is a membrane serine proteases. Okay, and there is cathepsin L. Okay, what they do is they are usually present on the host cell membrane. Okay, they just go and they cleave S1 and S2. Okay, so what happens when the S1 and S2 is cleaved? This S1 and S2 come here. Okay, S2 is here, S1 is here. Okay, this S2, it combines with a beautiful receptor on the cell surface. Okay, it is present abundantly on the alveolar, lungs, kidney and all that. What is this receptor? This receptor is something you have been reading from the first year of your medicine. This is ACE2 receptor. Okay, this S2 combines with this ACE2 receptor, and what is the final end product of all this? The virus gets bound to the cell membrane. Okay, now this S1, what it does is it causes fusion of the viral cell membrane with the host cell membrane. Okay, so there is attachment with the help of S2 and there is penetration with the help of S1. Okay, so spike protein is responsible for both attachment and penetration, and it is aided by PMSSR2, cathepsin 2, and it binds to the ACE2 receptors. Okay, so this is the basics of viral entry into the cell. Okay, how a viral enters into the cell. Okay, this basics is right now enough for you to understand the mechanism of action of these antibodies. Okay, the others we'll just see very rapidly with the help of the picture I showed above. Okay, here I'll go to the picture here. So in this picture we see that the SARS virus, it binds to the cell membrane with ACE2, and there's this TMPRSS2, and then there is cathepsin L. Okay, so this cell, this COVID virus, it enters into the cell. Once it enters into the cell, it is consumed by the lysosome. Okay, we think that getting it to the lysosome is good, but no. What it does is there's a release of the viral genome. Okay, this viral genome, it goes to the ribosome, and this viral genome is translated into proteins PPA1 and PPA2. Okay, this genome, it combines with the endoplasmic reticulum of the cell, and it produces certain substances. Okay, so what does it do? By replication, it forms nucleocapsid, spikes, membranium, and envelope. What are all these? Okay, these are all parts of the COVID virus. Okay, so these combine with each other, they assemble and bud, and they form a new virus, and this gets released. Okay, this is very basic. I really wish I could go in detail, but it takes a long time, and probably if you people are interested, I'll just take a class wholesomely on the pathogenesis of COVID, which is just so interesting and so massive. All right, so but pertaining to this antibody, we just need to know about the viral entry mechanism. Okay, so so the spike protein combines to ACE2 receptors, and there are certain binding proteins on the spike protein. This is known as receptor binding proteins, which bind to the ACE receptor, and this viral entry into the cell. We know the role of TMPR as a transmembrane protein serine proteases, and cathepsin L, and this ACE2 receptors. All right, so now what happens? Okay, we saw about the retinol receptor binding domains. So what scientists did was they developed antibodies to this receptor binding domain on the spike protein. There were 4 antibodies that are now in research: bamlanivimab, etesevimab, casirivimab, and imdivimab. Okay, so this bamlanivimab and etesevimab, they are used in combination, and imdivimab, casirivimab are used in formulation. This combination is approved here. Okay, remember, they are non-competitive. Okay, they non-competitively, they bind to the receptor binding domain. Okay, so what happens when they combine to a receptor binding domain? They prevent in binding of this receptor binding domain to the ACE2 receptor. It's so simple, right? It's so simple. So there's a non-competitive 2 antibodies combining with the receptor binding domain, and this receptor binding domain, it prevents this from binding to the ACE2 receptors. Okay, simple. Okay, so this is the mechanism of action. Now the doubt comes to my mind is, why is there a need for 2 antibodies? One antibody could do the trick, right? So why is there a need of two antibodies? So why a cocktail? Okay, so I read about it. So the concept is, there is something very special about a virus. Okay, especially a respiratory virus. Why are we so scared about influenza? Why are we so scared about SARS? Okay, these viruses, they're very clever. Okay, they keep mutating and all that, and what they do is, if we use a antibody that is targeting one specific domain, what this virus does is, in this domain, it simply mutates this domain. Okay, there's a simple mutation of this domain, and the antibody doesn't work anymore. They form a mutant variant. Okay, so the concept is preventing this or slowing down this mutant, so we target two domains. Okay, so the chance of this failing will be low. This is the basic concept behind an antibody cocktail. So an antibody cocktail doesn't necessarily mean only two antibodies. It can even contain in the future 3 or more antibodies, and this can be actually very effective future treatment of COVID. Okay, so what it does is it prevents the virus from entering the cell. Okay, so it prevents replication of the virus. Okay, so that is why we use it in early stages. We use it in mild and moderate disease. Okay, to prevent replication. But in cases of a already full blown virus in a severe disease, the use of this is very questionable. Okay, you get it? What it does prevents the SARS virus from binding to the ACE2 receptor. So it is useful only when the patient is actually developing the disease. It is not very effective in a full-blown disease. You have to remember that. Okay. So why a cocktail? It prevents mutation. All right. So the indication is a mild to moderate disease. You know why. And the dose, they tried a 1200 mg dose and a 2400 mg dose. They both found that both are like equal in efficacy. So we can see there's a 70% risk reduction. Okay, this is the phase 3 trials of this antibody. And the cost is the problem, 57,500. Okay, so as per the mechanism of action and pathogenesis, this does look like a very interesting and futuristic drug that is very specific for COVID. Okay, so it does look interesting, but the problem is the cost, and it's just been put into trial. It has not been tried on a large population, but it's very interesting. Let's see how it works. Yeah, thank you.